Okay, may I share my screen now?
Yes, go ahead, please.
And actually, yeah, there it is.
So everybody can see my screen, full screen?
We see your screen. It looks good.
Okay, great. Okay, thanks for having me here for this seminars. It's my great honor to meet many great peoples at Georgia Tech, and I'm going to meet more great peoples in the afternoon. So as Andres said, I did my PhD in Clemson University. So me and my wife, at that time she's my girlfriend. So we drove to Atlanta every month for Chinese food and grocery. So at that time, like Georgia Tech was my dream school for my postdoc actually. So I did apply, but didn't get a chance to come here. And then I went to Kevin Healy's lab and get to know Andres, and now he bring me back here. So like people said, like life came a full circle. So. Okay, maybe my short introduction work better in person. So in the next 45 minutes, I'd like to share with some of our newest result on engineering stem cell organoids specifically for drug screening purpose. So before we get into the real science, I'd like to take a moment to introduce my lab at the Syracuse University. We take the advantage of human-induced pluripotent stem cell as a cell source to investigate different problems in biomedical science and engineering. We primarily generated three cell types, mesenchymal stromal cells, cardiomyocytes, and vascular endothelial cells from iPSCs. We're using our mesenchymal stem cells for manufacturing applications, from tissue fabrications to cell manufacturings, and now we are going to a little bit into exosome manufacturings. And we use our endothelial cells to study the formation of vascular structures in 3D printed scaffolds based on a collaboration with the Solomon lab at Syracuse. And of course, cardiomyocytes is our main focus of the lab. And with that, we established two in vitro tissue models, including cardiac tissue model and the cardiac microtissue models. We're also exploring some new directions to using advanced AI technology to better analyze the contractile functions of our organoids and microtissues as well. So in this talk, I'm going to focus on our organoid model. I'd like to share some of our new results to see how this geometric confinement will affect the organoid formation and the functions. And towards the talk, if I have time, I don't think I can touch a little bit on the new project on the mesenchymal stromal cell differentiations, which we hope we can get into the field of cell and exosome manufacturing. So the advancement of human iPSC technology offers really unique opportunities in regenerative medicine, since we can differentiate many different cell types from iPSCs for tissue repair and regenerations. And specific for cardiac differentiations, we can derive cardiomyocytes with high efficiency. The differentiation protocols follow this developmental cell biology with a cell-free transition from pluripotent stem cells to the mesodermal cells, and then to the cardiac progenitor, and finally become the working cardiac muscle cells. So you can see this cardiac muscle cells can form a sheet-beating cell layer in the tissue culture plate, and they also express key molecular feature signatures of cardiomyocytes. But every time I show this beating video, I have to tell everybody that this is the best video we can get. If you see such beatings from other presentations, this is probably the best reading they can get as well. So everybody claims they have really high efficiency. So in our hand, and I also talk many people work on the cardiac differentiations. The efficiency is about like 60 to 70% of cardiac myocytes productions. So if you really want to get to like a high purity of the cardiac myocytes, you have to using the purification protocols. So, however, if we go back to the traditional developmental biology, what is important here is not just the simple cell-free transition from one cell type to the others, but also the complex tissue growth and the tissue morphogenesis occurring throughout the entire developmental process. Again, specifically to the heart development, you can see in the video on the right, is a cheek early embryonic heart formations. In that videos, the mesodermal cells migrate a long way to the center of the embryos and fuse into the linear heart tube. And this linear heart tube can loop and grow into the heart chambers. If we put this in vitro stem cell biology and in vivo developmental biology together, there's many questions ahead of us. And my lab particularly interested in two major questions, how the cell differentiate and the tissue morphogenesis coordinate to form complex tissue, and how we can create an in vitro model system to recapitulate this developmental process. And these questions bring us to the stem cell organoid technology. So traditionally, people can grow the stem cell aggregates in soft hydrogels. In most cases, it's matrigel, and induce the differentiation into specific lineage. And this 3D aggregates can differentiate and develop into organoids that can mimic the early stage of a developing organ. There are many labs has been developing so many different tissue-specific organoids, and my lab is primarily work on the heart organoids. Meanwhile, as a bioengineer, right, we are seeking a better approach to control the stem cell differentiation and organoid production. This including growing organoids within a synthetic hydrogel instead of a traditional matrigel. For example, packed hydrogel has been used for organoid cultures. Dr. Garcia has made a great contribution in this field. On the other side, microwell and the surface micropatterning techniques has been also used to provide a controllable way of to control the size homogeneities and the colony confinements. And these techniques can be also used for high throughput scale-up productions of organoids for different applications. And more recently, integration of microfluidic systems and organoid cultures create this organoid on chip platform for better control the dynamic organoid microenvironment, and also give us a method or opportunities to study the multi-organoid interactions. With all these technologies, the organoids has been used for different applications. For example, disease modelings. People use it for fundamental developmental biologies, and also used for like different drug screening applications. Specific to this micropatterning technique for organoid productions, which we use for our heart organoids, we can create a different geometry of the to pattern stem cell colony. The pattern surface will provide this geometric confinement to the cells, which can generate a gradient of biophysical input from the edge of the patterns to the centers. And that gradient will drive the spatial differentiation of the stem cell. Showing on the right is one, I think it's one of the best examples of micropatterning techniques for spatial stem cell differentiation and organizations. So by micropatterning the human embryonic stem cells into this circular shape and treating the cells with a key morphogen, BMP4, the EIC can differentiate and self-organize into this different layer of cells with trophoblast-like cells on the outside. And then next to it is an endodermal layer, and then it's the mesodermal layers, and then you have this ectoderm layer in the center of the patterns. This spatial organization recapitulates this germ layer differentiation during the early embryogenesis. So in my lab, we're using surface micropatterning method to grow our cardiac organoids. There's some more details in our published papers, but briefly, we grow a thin layer of a pad on the tissue culture polystyrene, in most cases, just like a 12-well plate or 6-well plate. And then we put a layer of PDMS stencil on top of this PEG layer. And the PDMS have a clear through hole. And where the hole is, we can use oxygen plasma to selectively etch away the PEG layer underneath. So there's a region without the PEG layer, and that region we can coat in with Matrigel and grow our iPSCs. After we grow our iPSCs, we just follow that traditional cardiac differentiation protocol to generate our cardiac organoids. So this is some like a surface chemistry I did when I was in Kevin Healy's lab. So basically, we can show where is the PEG layer is and where is not, and we coated the pattern surface with Matrigel. And the Matrigel can only deposit onto the region without PEG, so we can grow our human iPSCs into these specific patterns with a different size and shapes. We also show that iPSCs can maintain their pure potency after micropatterning procedures. So, and then we just follow this cardiac differentiation protocol developed by the Sean Palachek groups using 2 small molecules to modulate the Wnt pathway. The bright field image down below just show you what the pattern cells looks like during the cardiac differentiation. We show there's a morphology change before and after the cure treatment, and also before and after the IWP4 treatment. We start to see the formation of multi-layer cell aggregates after the cure treatment for the mesoderm induction. And the tissue start to condensate to the center after the IWP4 treatment when the differentiation is more towards to the cardiac lineage. So normally we can have the beating organoids starting from day 10 if we consider the cure treatment as a day 1, and then we run our endpoint measurements on day 20. So this is our cardiac organoids. I like to highlight 2 things. One is our organoids. They are generated from 2-dimensional iPSCs colonies instead of 3D aggregates that people normally use to generate organoids. And we think this is a 2D to 3D process that can represent certain level of tissue growth and tissue morphogenesis. The other things we like to highlight is the beating heart muscle is on the center top of the organoids, showing on the red colors, but the stromal cells showing as green colors is actually on the perimeter of the organoids. It's sort of on the bottom of the organoids. This representing the spatial organization of the different cell population within our organoids. So we did more staining to show our cardiomyocyte markers of cardiac troponin T, troponin I, myosin heavy chain, sarcomere actinin. All these markers indicate that the cardiomyocyte is on the top center of the organoids. And then we also staining for smooth muscle cell markers of smooth muscle actin, troponin, SMN22, and vimentin, they are shown more on the outside of the organoids. The bottom row of the image are the confocal image of the cardiac markers. Through this staining, you can see the cardiomyocytes not aligned in our organoids, which is quite different compared to the other cardiac microtissues that in the field, which people try to use an engineer approach to create this aligned cardiac tissue for more like physiological relevant functions and response. So the image on the left here is a 3D reconstruction of the confocal image of our cardiac organoids, which show the sort of a 3D dimensionality of our organoids. However, you can see there's a huge black, like a dark, basically black region in the center of the organoids. And that was because the limitation of a confocal microscopy for their like a low light penetration for deep tissue imaging. So, and then we go into this two-photon microscopy, which show on the right of the slides. And you start to see some internal structure of the cardiac organoids. We do see some internal region showing there's a no cell nuclear, indicate that region inside the cardiac organoids have no cells. There's a paper published early this year saying people can generate microchambers in the heart organoids. But however, we don't think we create a true chamber in our organoids because if there's a chamber, eventually the organoid is going to collapse because all this cardiac contraction will condensate the tissue into a more like aggregate structures. So we think that the reason we can retain a region without the cells is because that region maybe was filled up by the extracellular matrix produced by the supportive cell types. In development, people call it a cardiac jelly, which is filled in the linear heart tube. And I think something happening in our cardiac organoid as well. We haven't really further characterized our extracellular matrix production yet, but that's something we are planning to do. So to characterize the structure of the organoids, we can measure the area of the cardiomyocyte staining and calculate the area ratio between the cardiomyocyte staining over the entire pattern size. This can give us an estimate how well the cardiac differentiation based on the cardiac muscle coverage on the organoid. We can also measure the height and the width of the organoids, which can give us an estimate how well the tissue grows and the 3D organoid formation. So we generate the cardiac organoids from circular pattern with different size. We see from a 200 micron organoids showing this high area ratio, but much higher data variations. So in general, we can now get a consistent organoid production from 200 micron patterns. And we found the organoid from 400 and 600 micron circle patterns have a relatively lower area ratio, but larger organoid height and much better consistency in the organoid size and also cardiac differentiations. Based on the organoid width, we see the organoid size are now scale up with the increase of the pattern size. So especially when we move to the larger patterns, such as 800 or 1000 micron circle patterns, we see the cardiac differentiation have a tendency to follow traditional 2D cardiac differentiations that we see in the tissue culture plate, instead of forming a nice 3D organoid structure. So this data tell us a little bit how this initial pattern geometry will affect the three-dimensional structure of the organoids, and how that will affect the organoid productions as well. So to characterize the cardiac organoids functions, we're using GCAM reporter IPSL's line for calcium handling analysis. So basically, we can perform bright field video recording for contractile motion analysis, and then we can perform fluorescence video recording for calcium analysis. From this analysis, we can extract a series of parameters to quantify the functions, which is showing like in this waveform. For example, we can have the parameters of contractile velocities, relaxation velocities, calcium decay time, and also like upstroke calcium increase time. So this is a busy slide. I'm not gonna like explain this to you. You know why? I'm not gonna explain this to you in the next couple of slides. But the idea is to analyze so many variables, so many graphs, and so many statistical bars. It's hard to make a conclusion. You compare so many different variables for different organoids with different size, it's just hard to extract useful information. So since I started at my lab and SU, I start to ask the questions how we can better interpret this data, how we can explain this data, and what's the key information that is buried in all these different graphs. So what we want to do is to have a better way to analyze and visualize the data using some advanced tools in data mining and data analytics. So we put so in this slides we put together these parameters for organoids generated from different pattern sizes in this heat map, and we also show 10 different parameters we can analyze in this data analytics approach. So we further analyze how these parameters can correlate with each other. So we can generate this correlation matrix. For example, from this correlation matrix, we can see there's a high correlation between this pulse duration from calcium analysis and the peak-peak interval of contractile motion analysis, which means if we perform, even though we perform two different analyses, the data can highly correlate to each other. We observe this. We also observe that the B rate is negative correlate to the B duration and the calcium decay. We also find there's the area ratio of the organoid morphological analysis is a positive correlate with the contractile motion analysis, which indicate that the larger organoids contract stronger. So basically, using this correlation matrix can help us to establish this structure and function correlations for our cardiac organoids. And next, we want to see whether we can integrate all these parameters for each organoid and analyze the correlation among different organoids. So we're using this t-SNE plot to map all the organoids we have collected data from. So basically, you can see each dot showing in this map is one organoid integrate with 10 different measurements from previous slides. So we observe that the larger organoids from 800 or 1,000 micron patterns, they are clustered quite well in this t-SNE plot. But smaller organoids with a 200 and 400 microns pattern size showing this separation in the t-SNE plot, which indicate that the larger variation in the organoid's functionality for the organoids with a smaller size. To further dive into each parameters, we can see that this divergence of smaller organoids is mainly result from the difference of the beat duration and the causing decay. So this analysis can help us to pin down the fundamental key difference between smaller organoids and larger organoids, which can help us to further optimize organoid productions with consistent functionalities. So, and then to further determine the cell composition of our organoids, we run the single-cell RNA sequencing on day 20 organoids. And as expected, we found our cardiomyocyte populations and the stromal cell populations containing both the fibroblasts and the stromal and the smooth muscle cells. And surprisingly, we found a large population of cells that actually came from endoderm, showing signature genes related to gut epithelial development and going to early hepatic lineage for liver development. So, and this is like we are quite exciting with our new results. And then we see this publication came out in last month in the Nature Biotechnology. They also showing this hard forming organoids having this endodermal foregut lineage differentiations. So basically, they claim in the cardiac organoids differentiations, the differentiation protocol will drive both the cardiac mesoderm and the cardiomyocyte differentiations, and at the same time, the foregut differentiation into both lung and the liver lineages. So we are frustrated a little bit by somebody published the data before us, but then we find that there's a new data from our single-cell RNA sequencing experiments showing that by changing the geometry of the patterns, we actually can tune the cell compositions. Showing this graph, we see with the 200 microns patterns, we can generate the cardiac organoids with more stromal cell populations, but with the larger organoids from 1,000 microns patterns showing the more favorable differentiation into the gut lineage. And these middle-sized organoids with 600 micron patterns actually show primarily cardiac populations. So like one thing I like to highlight here is for the cardiac populations, basically looking at these cardiomyocyte populations, there are actually two separated clusters of cardiomyocytes showing in the 200 micron organoids as the blue colors, but in the 600 and 1,000 microns organoids showing as the green colors, which indicates that the different pattern size will change the phenotype of the cardiomyocytes, or maybe a different pattern size will induce the different maturity of the cardiomyocytes. So this is like a slice of our ongoing work. So basically, we're just taking the organoids at the different days during the differentiations. For example, day 0 is just IPICs. Day 1 is after cure treatment, after the mesoderm inductions. So basically, we are trying to mapping all the cell types we can find during the entire organoid productions process. And showing on the right is the different cell types. We are still need to further dive into developmental biologies. And then we also establish a new collaboration with Ben Koskov lab at the Cornell Universities and try to using their more advanced bioinformatics tools to help us to see more subtypes cell populations in our single-cell RNA-seq data. So now I'm gonna switch gear a little bit and talk about the application aspect of our organoid model. So taking drugs from pregnancy is always a guessing game because we are so afraid that the drugs will have side effects that will influence the healthy fetal development. However, nearly 40% of pregnant women in the US still receive a drug which the safety for the fetal development has not been well established yet. So this comes to a very difficult and a crucial decision for the expecting mother about their pregnancy medications, especially for the women who are suffering from mental disease because the safety for psychiatry drugs during pregnancy are not well studied and established. When we talk about this drug-induced teratogenicity, we cannot ignore this thalidomide tragedy in the history. So this drug was first developed to treat nausea, and then the pregnant women taking it to alleviate the morning sick. In the late 1950s and the 1960s, more than 10,000 children in 46 countries were born with deformities as a consequences of thalidomide use. And unfortunately, only 50% of the babies survived. So this tragedy pushed the FDA to establish this drug category that can use to regulate the drug prescriptions for pregnancy women. From the category, you can see any drug from C to X will potentially have adverse effect on the fetal development. And unfortunately, a lot of drugs we use for treat diseases or syndromes fall in this category. However, in 2016, FDA decided to remove the drug category system due to unreliability and lack of evidence from human-related studies. So one major reason of this unreliability came from this assay systems that for the embryotoxicity screenings that pharmaceutical company has been used for decades. Here's the 3 assays, including a whole embryo assay based on rodent or zebrafish, micromass assay, and the mouse embryonic stem cell assay. There's no human assay available. And more important, only the whole embryo assay provide us this morphological scoring that evaluate whether the tissue or organ can develop into the right form under the drug exposure. So what we really need to establish is to create a human model for embryotoxicity test which have the capability for morphological scoring. So taking our advantage of cardiac organoid models, we test many drugs from these different categories with different potential of the embryotoxicity. Just be simple, you can think with the increase of the category from A to X, the severity of the embryotoxicity level is increased. So for the time, I'm not going to show all the results for every single drugs, but in the next 15 minutes, I'm going to try to focus on some key interesting results we observed from our drug screening test. So first, we test our category A drug, which means there's no effect on embryo development. We didn't see any difference on the area ratio across the different dosage on the organoids. And we also, but however, we do see that the drugs have an effect on the B rate. One reason because this drug was prescribed as a sleep aid, which can reduce the muscle contractions. That's the reason we have a lower heart rate that can help us to go into the sleep. And these drugs do not affect the height or the width of the organoids, indicating that the organoids can grow into the right 3-dimensional structures. So then go to the totally the other way. We test this thalidomide. which is a category X drugs, we find is at the highest dosage, it will significantly diminish the cardiac differentiation. And surprisingly, the thalidomide does not affect the contractile functions from the organoids, at least from the motion tracking analysis. But when we perform the confocal image of the organoids, we can see the 3D morphology of the organoids was significantly impacted by the drug exposure. So we found even at a low dosage at 1 micromolar, the thalidomide could significantly reduce the organoid height and limited organoid developing into the right structure. So I also like to highlight this drug. This is a radionic acid. There's 2 versions of the radionic acid, tretinoin and isotretinoin. The difference is only 1 chemical bond rotation, which make one drug is a category D and the other one is a category X. So when we test these 2 drugs using our organoid models, we found the category D drugs with less embryotoxicity and can generate cardiac organoids at 1 micromolar. But the category X drugs, again, only 1 chemical bond rotation, cannot generate organoids at the same concentration. More interestingly, we know at the highest dosage of the retinoic acids, there's no cardiac differentiation, but we still can find the formation of a giant 3D tissue structure, which is even larger than the normal cardiac organoids. This told us there's no cardiac differentiation does not mean we cannot get a large organoid tissue formation. So last, we also compare our result with zebrafish embryo models. This transgenic zebrafish model has reported on the melting light chain, so we can analyze how the drug will affect the looping of the heart tube in the zebrafish. For the 4 drugs I mentioned here, we found a consistent result of the sleepy 8, that's the category A drug, and radionic acid between the human organoid models and the zebrafish embryo models. However, the thalidomide exposure did not induce a significant impact on the zebrafish model, which highlights the need of a human-based assay for the embryotoxicity. To summarize my talk on the IPS cardiac organoids, we established a human development heart model on the cardiac organoids. We investigate how this geometric confinement will affect the structure-function relationship of the cardiac organoids. And this developing heart model can be used for the assessment of the drug embryotoxicity and safety screening for pregnancy medications. So I think I'm just stop here. I don't think I have time for the mesenchymal stem cell part. I can take questions, and for the people interested in the mesenchymal stromal cells we differentiate in the lab, we can talk more in our meetings. Or if we don't get a meet, you can just send me an email if you're interested in the mesenchymal stem cell that we can differentiate from human iPSCs. And thank you very much for your attention. I'm happy to take questions.
Great. So virtual clap. Thank you very much. That was a great seminar. Please post your questions in the chat. Zhen, we have 3 questions, so I'll go through those.
Okay.
So the first question is from Professor Mantalaris. Could the higher variability observed in the 800 to 100 micron organoids be due to potential mass transport limitation? I think he's referring to the first part of your presentation.
Yes. So data variations we see like among the organoids, right? Like especially when we do this t-SNE plot and try to see how all the organoids can cluster each other, right? We see in most cases the larger pattern size, the organoids, they can cluster quite well in the t-SNE plot, which means there's a higher consistency in the larger organoids compared to the smaller organoids. And so I think like if we couple the functions result with the single cell RNA sequencing result, from the single cell RNA sequencing result, we show there's a more progenitor cells and the stromal cells located in the smaller organoids, which means the smaller organoids either in the phase which now they fully differentiate, at least at the day 20 mark, or they are locked in the stage, they can now fully differentiate into end fully differentiated cardiomyocytes or the gut lineages. And I think that like a progenitor stage will create a lot of like variations in the functionalities that we can measure at the endpoint measurements. So,
okay. And I was going to say, Saki's followed up with a sort of a more general question.
Mm-hmm.
What is the influence of mass transport in the results? Because you have these very large cell aggregates, right? So
think about like the organoids more like a dome shape, right? And our organoids never really go above 300 microns. So if you think about the core is the center, it's never really, it sort of reached the limit of the nutrient penetration, about like 200 microns nutrient penetrations, right? So one thing I think that the tissue can now develop even longer or larger is because the limitation of the nutrient transport. So there's only a couple of cases we see that they can grow a huge like organoids that actually from our radionic acid treatment, we cannot really see inside yet, but maybe there's a necrotic core developed in the center of in the organoids. We cannot just see. But for like smaller organoids, I don't think there's a nutrient transport issue occurs.
Okay, thank you. Okay, so we have a question from Hannah Song. 3D cardiac organoid generation looks very interesting. In the heart, cardiomyocytes and cardiac fibroblasts arrange almost like alternating each other. In the organoids, it seems like they differentiate and proliferate separately. Any idea what kind of signaling is involved in these two different development-like processes?
Oh, we haven't really go into the signaling part, but I can tell you a little bit like some initial thought why we have like stromal cell like more on the bottom. So basically, like our organoids is now cell floating in a matrix, right? They are differentiated from micropatterned colonies, which means the organoids basically is stuck on the bottom of the substrate. So, and the substrate is very stiff. And then they need some cells that can holding up the entire tissue and stick to the bottom. And that was the stromal cells. So basically, the bottom layer is the stromal cells that can support the entire organoids. And then on the top, it's much softer, the tissue much easier to contract, to move, and And that for the cardiomyocytes, because they need a contract, it's much easier to differentiate on the top of the organoids. But of course, I only take this more like a mechanobiology aspect of these spatial differentiations. There should be some signaling behind this spatial differentiations. But I can tell you, first thing is when we do some staining at the day 4, when the mesoderm and endoderm start to co-emerge on our organoids. Actually, the mesodermal cell and the endodermal cell, they are mixed together at day 4. But when we do the staining at day 20, again, using the cardiomyocytes plus other like endoderm markers for liver and some liver markers, so the spatial differentiation occur showing up on the day 20. So I don't know what's happening from day 4 to day 20. So at the beginning, so the endoderm and the mesoderm was mixture, and they find a way to sort them out in that 16 days to the final product of the cardiac organoids. That's something we are really interested to looking at. And we are currently, as I mentioned, I collaborate with Koskov lab and try to using some bioinformatics method to try to see this ligand receptor interactions between different cell types that probably can give us better ideas how this endoderm and the mesoderm crosstalk each other and to create that spatial differentiations we see at the day 20 organoids.
Okay, so Ali posted the following question. Beyond the cardiac formation and beating frequency, could you leverage your earlier data showing endodermal differentiation in some organoid sizes to study drug effects on this development or how it interplays with cardiac development?
So that's really great questions. That's something we really want to do because in development, right? Heart and liver, they are co-development. Heart is slightly early than liver, but basically like a liver, like a heart send a lot of signaling, BMP signaling to the liver, and the liver like pay back a lot of FGF signaling to the heart. So there's a lot of crosstalk between the heart and livers during the development. There's something we really want to do and looking at. So one thing I like to highlight is also from our data I didn't show here. There are some population of the cells in the endoderms. They're actually expressing some genes inhibit the cardiac differentiations. So I think like in our this like organoids, they have to find sort of a balance between the mesoderm and endoderm. So they promote each other at the same time. They also inhibit each other. I cannot say each other because our protocol is primarily drive to the cardiac differentiations. So maybe the emergence of the liver, emergence of the endodermal cells will play a role, try to slow down or reduce the percentile of the cardiac differentiation in our organoids. So, so like if I can using some magic way that can totally remove the endoderm like a cell differentiations, maybe I have even better cardiac contractile functions from our organoids as well.
Okay. So another question. The cardiac organoid 3D model is very useful for drug screening. We are developing drugs targeting tropomyosin isoforms such as TIMP-3.1, which regulate the functions of specific populations of actin filaments. Since TIMP-3.1 is not a muscular isoform, do you think your model can be used to test these anti-TIMP-3.1 drugs to validate that they have no impact on cardiac functions?
I'm sorry. I think I lost a little bit. I didn't quite get the half part of your.
So they have drugs that target tropomyosin isoforms such as TMP 3.1. I don't know how to pronounce that. But that tropomyosin is not a muscular isoform. So do you think you can use your model, your cardiac 3D model, to test the drugs to validate that they have no impact on cardiac function?
I think yes. There has been many different models that can be used for testing drugs or the disease modeling aspect. So at the very beginning talk, I do show we have another model. We call like micro tissue models, which try to recreate the cardiac muscle that's more similar to the heart, which is highly elongated and uniaxial contractions. And that platform, we can measure the force as well. So for the organoids, we can now measure the force. So, so I think like for a lot of like cardiac responsive drugs, normally we go to that model instead of organoid models. But I'm happy to let the conversation rolling to using our other models to give a test on the drugs, how the drugs will modulate the contractile functions over the cardiac micro tissues.
Okay. The next question is from Professor Eric Dreden regarding translation of your teratogenicity assay. I'm curious what future studies would be required from confirmation. Human trials, but that probably would be a difficult sell.
So you mean for the embryotoxicity studies?
I think it's for for your assay of teratogenicity.
So so currently, like this is my like R1 grant. So basically, my R1 grant is just try to using this model to establish like a biostatistic model. So basically, if you have a new drugs, we can feed in the model, measure the data using our cardiac organoids and feeding in that statistics model, and then the model can tell you what's the risk level of embryotoxicity. So and this is how pharmaceutical companies using their mouse model in their research. So it's I think like in terms of this how's the progress, right? I think people should be take like one step forward, but now like directly go to the endpoint to humans to compare. So I think like for a near future, what I really want to do is just compare so many different models for embryotoxicity, not just the physical model and also the computational models with that risk classification systems to determine which one has the better predictivities, right? Which have the more precisions on the embryotoxicity screening. So I just don't think eventually like this is the way people can do a lot of human trials with the pregnant women, right? So for vaccinations, pregnant women should be excluded. So it's just not a way to do it.
Okay, and we'll take the last question is from David Wolfson. Do you think it would be possible to leverage your single-cell RNA-seq data and drug screening platform to find differences in cardiac chamber or phenotype-specific responses to various cardiac drugs?
So yeah, that's I think like the single-cell RNA-seq open up a lot of possibility for us. So By the other side, so this experiment is very expensive. We are now planning to do all the screening or transcriptional analysis using single-cell RNA-seq. So we are currently actually doing a lot of bulk RNA-seq as well for drug screening applications. We want to see the drug exposed organoids and then control organoids. So so we can see how the drugs will affect on the transcriptome profiles. So if we need really see how the drugs will target to really specific cell types, right? Single-cell RNA-seq is the way to go.
Okay, great. That was perfect timing. My chime just went off for 12. So everybody, please join me on thanking Professor Ma for a wonderful seminar. And have a great day. Remember, as indicated in the chat, the seminar will be available in the IBB website. And again, I wish everybody stay safe. And Sheng, I'll see you later this afternoon. And I think Rose may have something to say.
I would rather ask everyone to email me if you're interested in the link. It will be made available this afternoon, and my email address is listed there for you.
Okay, thank you. Okay, thank you everybody.
Thank you everyone. Thanks for coming.
Bye. See you in a little bit, Sheng.
See ya.